central importance of adhesions if you're studying mechanobiology, because that's where the contact between the cells and their surroundings kind of lies, and it's more complicated than one might think. So there are lots of adhesions in real life. You hold on to something, that there's an adhesion. There is a slip bonds or really clamped sort of bonds. There are lots of different types of bonds. The one I like kind of thinking about when I think about focal adhesions is like a Velcro. So where there is a soft surface and there is these little plastic hooks, they're tiny ones, and it looks simple, but it's pretty complicated. And each hook is not, as in terms of numbers, proportional to those little fibers on the other end. There are less number of hooks, lots of little fibers. And if you put the two surfaces of Velcro kind of next to each other, really next to each other, at some point they'll start sort of interacting and the formation of bond happens. And after that, you kind of have to press on it, right? So if you press, then the Velcro is nice and tight, and then it is harder to kind of sort of unbond that thing. And unbonding a Velcro is not straightforward either. If you give it to a child, they might play with it for a few seconds. As a grown-up, I know you have to kind of unwrap it, so the shear won't work or the normal force won't work. You really kind of roll it over. So it kind of shows you the complexity of the nature of different types of adhesions and bonds that can exist, and that can change with many number of things, like what kind of force are you applying. This is a force sensitive bond, the Velcro. Like if you don't press on it, it won't adhere very nicely. How much surface is overlapping, that also depends. If you just have not very nicely overlapping number of sort of fibers, number of hooks, all of that will determine the strength of the bond and how it will function. So keep that in mind in terms of cells, what we want to talk about. I'll go next here if I can. Okay. So in terms of cells, what we're going to talk about, this is a cartoon that I like want to talk about here is the ECM, which is a complicated story in itself. There are lots of different types of ECM. So you already know some of this, and you might get to learn more this week, next 2 weeks. ECM is on the top layer, there are proteins. There are lots of proteins, collagen, bunch of different kinds, fibronectin, laminin, matrigel, which is a mess of all these things. So there are lots of different types of proteins. And proteins themselves are not really just floating, they're attached to some kind of a matrix, which can be elastic, viscoelastic, lots of different types of mechanical properties. But mainly, we'll just talk about the ECM as sort of a ligand. So you can think about it as the little hook on the Velcro. And on the cell side, there are integrins, which are the real receptors. So you can think about that little fiber thingy on the Velcro. And there are lots of them. They're moving around in the cell membrane, and they go and make the first bond with the ligand, which is the ECM protein. That's not it. So in terms of Velcro, that's it. There is nothing beyond the sort of the little soft fibers. In terms of cells, there is a lot more there. That is just the initiation of the bond, and that complexity is something I'm going to talk about today. Then inside the cell, there are these other proteins, focal adhesion proteins, that come and sort of stabilize the bond. And then that bond, it doesn't just exist for the sake of existing. The bond attaches to F-actin filaments, and then actin filaments. There is downstream signaling biochemical processes, which we call mechanotransduction, transduction of these physical adhesions and that signal into biochemical processes that drive actomyosin contractility, which is another story, which I'll get into in a second. And it is all coordinated. So the picture in the right sort of makes that point. So if you look at these green little long sort of structures, that's a vinculin, one of the proteins in focal adhesions. And I'll talk about again why vinculin is a popular one and what is the importance of phosphofACT, which will come later. The main point here is that actin is inside the cytoskeleton. It's one of the cytoskeletal proteins. It's highly kind of spatially sort of aligned with the focal adhesion. So there is some sort of connection, at least physical connection, not just like the bond, but correlation of how the focal adhesions are laid out and how the actin fibers are kind of laid out along with it. So they go hand in hand. So if the actin fibers break, then the adhesions break, and if the adhesions break, then the actin fibers break. And a lot of work has been done in that, establishing that connection. And it remains sort of a mystery, not mystery, I shouldn't say. It's not a highly tenable problem to decouple. Like, it's sort of a chicken and egg problem. If you don't have forces, then adhesions sort of go away, and if you don't have adhesions, forces go away, and hence these active myosin contractility. And there is a lot of work still being done to kind of decouple those things. So it is it looks easy, but sort of to establish the relative contributions of these two things in mechanotransduction is an open area of research. Going forward, I tease the sort of today's lecture as focal adhesions being the central sort of component of mechanobiology, because the lecture is about that. I'm going to make a big deal about that. But in the context of mechanobiology, what does it do? So I said earlier, a cell kind of sensing any surface, you have to it can't sense the surface without adhering, and that happens with focal adhesions. What happens after that? So you made the adhesion, sensing happens, then there is mechanotransduction, conversion of these mechanical cues, which are coming from the extracellular matrix or the environment. And what are these mechanical cues? That's also a very loaded term. So if you read literature, you say something is mechanosensitive. What is mechano? If you are in the stiffness varying kind of field, you say stiffness is mechanics, or which is extremely naive. It's like saying gene expression is biology, right? There is a lot more to it. So if you're really studying mechanobiology, think about what are the mechanical cues, which are many, right? So there is statics, meaning forces or stresses or shear and normals. All of that is whatever you learn in solid mechanics. Then there is kinematics, meaning things moving, displacements, velocities, acceleration. All of that is kinematics. Then there is material constitutive behavior, which a very small subset of that is stiffness. Other than that, there is viscoelasticity or swelling or osmotic pressure, all of those things. Those are actually forces, but the constitutive behavior, hyperelastic, linear elastic, nonlinear, and so on. And the fourth thing I should have added, which I didn't add here, is boundary conditions, which is a mechanical cue as well. Is the cell confined or not confined, or is it elongated or in a 3D matrix or a 2D matrix attached or not attached? So all of that is mechanics. So all of those cues individually or together can provide these kind of mechanical cues, which lead to lots of biochemical responses. If you're a biologist, you know that part, I think, really well. If you're a mechanics person, then you know the mechanics part really well. And the goal of, I think, mechanobiology is to be educated about both of those sides. Where do these forces and mechanics come from? And one of the exercises you can do, I guess, is to read different papers and see where the mechanical cue is. Which bucket does it lie in? Is it statics, or is it kinematics, or is it material behavior, or boundary conditions? And if you start looking at those specifically, you may find different sort of diversity in those mechanical cues. And here are some examples. I mean, endothelial cells, at the blood vessels, there is, of course, shear due to blood flow. So shear falls, I would say, in the statics category, forces and such. Blood pressure, also forces, stretching of the blood vessel walls is both kinematics and sort of forces. Rises in tissue stiffness, that is more the constitutive behavior changing. It's just stiffness is also a very naive term, I would say. It is what modulus, Young's modulus or world bulk modulus or shear modulus or viscoelastic, then you have these viscoelastic constants. So stiffness is fine, it's generally Young's modulus, but you can think it can be more complex than that. I'm digressing a little, I know, but just to show you the complexity of this. In terms of mechanotransduction, there is also sort of steps within it. First is sensing, mechanosensing. You have the mechano part, sense it. Then there is transmission. Whatever the sensing did to the cell, how is it transmitting within the cell or across cells? And what is the response? So sensing is one thing. If the sensor is not responding, then there's no point in sensing, right? Even if there are downstream cues, if they do nothing, there is no point of construction or transmission. So all three separately can be can be their own areas of research. First, the mechanosensing part. In a very rudimentary way, I would say sensing is making of these adhesions. Then there is contractility, forces being generated, and these cytoskeletal proteins sort of orchestrate this force generation. And I won't talk about that because I know there'll be other lectures about that. So going through this transmission part very quickly is that it happens very quickly. So if you, there is one experiment done here, which I've provided citation. There is a bead attached, magnetic bead onto the surface of the cell, and bead is pulled with a small force, and there is this calcium dye inside. And it shows on the right is that there is a very quick sort of uptick in calcium signaling, and the point is not the calcium part. Point is how quickly it transmits throughout the whole cell. So transmission is really fast within the cell by a various number of mechanisms. Again, those are not the highlight of this lecture. The second is sensing, which happens in sort of minutes. This is the part we will talk about in greater detail. So here, briefly, so focal adhesions are formed by these integrin receptor bond. So here is the cell is sort of upside down. The cell is down here. The substrate is there. And the receptor ligand bond is sort of made. And then there is this tailin protein. There is vinculin. There are hundreds of others, tens, maybe over 100 proteins that come and sort of stabilize this bond. And after that, this bond, once it's stabilized, attaches to the effectin fibers that then generate the force. And the force generated then stabilizes the bond. And this feedback is something we'll talk about in more detail. So in a nutshell, this is the sensing part. The reason why we call it sensing is that adhesion forming by itself is not sensing. But the cell responding to it, okay, there is an adhesion formed. Now I do something, which is in this case actin myosin contractility. And that itself is then responding, and in terms of focal adhesion maturation is the main sensing part. So if I were in the room, I would be able to see your faces, eyes glazing, and I might pause like, okay, is this making sense? Are there any questions? But since I'm not in the room, I will pause. It's been 5 slides. Are there any questions so far, or should I keep going?
I have a question. Can I ask a question?
Just yell it out.
Okay. So you mentioned about how in order to remove the cell adhesion or the focal adhesions, it's kind of a Velcro. So if the cell is being peeled from the ECM, the cell need to make sure that the focal adhesions are the ones that are broken, not the link between the focal adhesions and the actin, right? So
yes.
Can we say that the bond between the focal adhesions and the vinculin complex with the actin is much stronger?
Yeah, so that is, so I will actually talk about that in a second. So in my, I'll give you a short, long answer is that one of the most striking things I saw when I started working in cell migration, I changed from PhD to postdoc, I changed from being a sort of modeling person to experimental, is that when I saw my migration video for the first time, I thought I can work on that one video for my whole life. The reason being is that as you were talking about this cell being peeled, migration is a very fantastic example of that. So cell does that continuously. And the different types of cells do this sort of peeling or sticking and slipping motion in different ways. So there are fish keratocytes, so that kind of roll, like if you take a balloon and just plop it on a surface and just push it, it just rolls along. And even in that, there are bonds being formed in the front and broken in the rear, but they're in coordination. But slip sort of migration cell kind of holds both ends and kind of forces itself and moves forward. And in those videos, what I saw is that little parts of membranes are kind of floating behind. The cell is surviving, but as the rear adhesion is breaking, the part of little membrane kind of breaks with it. So that it hasn't been investigated kind of thoroughly, but what I would say to your question is that the integrin ligand bond sometimes can be pretty strong, and the part of the membrane that is attached to sort of these integrins sort of breaks. So focal adhesion bond that is here, vinculin, tailin, that might be stronger than the ligand sort of receptor bond.
Okay. Thank you.
Yeah, you're welcome. And so going back to, before I go back to adhesions, I want to kind of note the mechanoresponse part, which I think is, as you start to read mechanobiology, you'll realize it is the most complex and understudied analytical part, which is very hopeful for the field of mechanobiology, because you'll keep studying this. I think field is very much in infancy, and people will keep studying it for decades, if not longer, because it is so complicated. What happens after this mechanotransduction? There are a million things that happen. Is that people have talked about epigenetic landscape changing and gene transcription, and there are of course thousands of those, and they all have different roles to play in many number of developmental and diseases processes. Some last over minutes and hours, some last over days and weeks, some last over years, some are heritable even, supposedly. So I think this mechanoresponse part, and I'll let you kind of read this slide on your own, but main point is it's really complicated, and I find it very exciting in how many different ways mechanics can induce mechanotransduction and then have its response laid out in many of these processes. For instance, in my own lab, there are different projects. One is about development, where let's say in gastrulation, mechanoresponse is manifesting itself in formation of embryos within, let's say, weeks or days, and maybe a week or so. Or in cancer, there are these tumor formations that happen over weeks or months, and they may have secondary metastases that can come about in years. So all of these can be considered mechanoresponse. They're not just mechanoresponses, but mechanics does play a role, which has been shown. So I'll move on from this slide, because that's not the main point. Go back to focal adhesions. Let's revisit this idea, because we do want to kind of understand this in detail. So there's the ECM, there is the integrin, and there are these focal adhesion proteins in these different colors of green, and effectin fiber sort of, in this case, sliding over. So initially what happens, so imagine like the Velcro example, if you have the top surface and the bottom, and you're just sliding it over, the speed at which you're making the contact will determine whether the Velcro kind of sticks or not. If you go really fast, none of them have a chance to kind of stick to the hooks. And if you go really slow, without even the normal pressure, there is a higher likelihood the Velcro will stick. So you can imagine if this actin fiber is going really fast, the adhesions may not mature. So the ones that do not mature, even though the receptor ligand bond is made, those are called nascent adhesions, meaning there's a promise of adhesion, but the conditions didn't arise that it will become something else. So these are smaller, let's say less than 1 micron diameter. Then they become sort of focal complexes, meaning multiple of integrin ligands come together and they make a little focal complex. The reason why it's still not an adhesion is that adhesion proteins have not stabilized it. Then if these focal complexes come together, actin filaments also attach, and focal adhesion proteins also kind of come on top of each other, then becomes a focal adhesion. And this can be understood in terms of motor or clutch type models. So this focal adhesion serves as a clutch, and these effectin myosin activity can be understood as a motor, because these are happening due to moving actin bundles and myosin heads. And there are lots of different sort of going further in this clutch idea. So there's a typo here, it says catch and slip bonds. So there are catch bonds and slip bonds. So let me put the text here as well. Okay, so on the left, if there is a soft ECM or the force generation is kind of low or slow, meaning cells are slowly migrating, or force generation is low, which can happen on soft ECMs. Then the effectin fibers are moving fast. There is the focal adhesion lifetimes are small. So the only way the bonds are being formed is not by stabilization. So bonds are breaking, and when they break, they expose the previously bound receptor site is now open, and the new ligands have a higher chance to bind it. So the bonds are being formed by the virtue of them being broken at the same rate. So it's called adhesion turnover. And my previous sort of example of these fish keratocytes rolling over a surface requires slip bonds, because they don't want to make the adhesions too strong, because it'll make them much harder to break. But at the same time, not too weak, so they can't provide traction just right, these slip bonds. So the turnover happens, meaning bonds break. By breaking the bond, other surfaces are exposed, then those bonds are formed. So there is a sort of a more coordinated formation and rupture of bonds. That is the slip bond dynamics. Catch bond dynamics is a little bit more like the Velcro example, where first the adhesions are formed, but they won't stabilize until forces are applied. So when the forces are applied, what do the forces do? So if you have a bond formed, and it's kind of counterintuitive that if you apply the force on this bond, it will stabilize. At least this tension force. Though what happens is that there are other proteins, tailin, for example, that open up. So they exist like this, and you apply a force, they open up. And when they open up, there are other binding sites for other focal adhesion proteins. They come and stabilize those. So that's what the catch bond dynamics. But if you keep applying the force, in the end, it'll rupture. So catch and then rupture. So these are two sort of examples of these bond dynamics in focal adhesions. On the right, stiff ECMs or higher forces exhibit these both types of slip and catch bonds. On stiff ECMs, there are lots of catch bonds in the back, slip bonds in the front, and which I'll get to in a second. Any questions about this? Okay. So here, these are sort of temporal and spatial, I would say, steps in adhesion maturation. So in the beginning on the left, these are nascent adhesions. There is ECM, integrin, receptor, tailin. This is made, so this is still a nascent adhesion. But then after some focal adhesion proteins come and sort of aggregate on this adhesion, and still a focal complex, not really adhesion, become a complex which is ripe for making adhesion. Adhesion really happens when effectin fibers attach to these focal adhesions and start generating forces. And they form adhesions, meaning bigger complexes, sort of in two different ways. One is the same adhesion can keep getting bigger by applying forces, more proteins getting stabilized, or these little complexes can come together, come next to each other and aggregate, and that will make a bigger adhesion. So those both have been shown previously. Next is what happens after these adhesions are formed. So they act as mechanosensors, meaning I already said when they generate forces, these cryptic binding sites open and mature the adhesion. But there are other downstream signaling pathways. The most popular one is Rho, which Rho GTPases that get activated, and there is because of this integrin signaling. And the last of other, this JNK, PI3K, some of these are oncogenes as well. They are also related to Rho GTPases. Essentially, they upregulate actomyosin contractility, which increases forces, and then forces in turn increase adhesions. This is sort of the sensing and response part that I was talking about earlier. So this is the sort of basis of mechanotransduction. All right. So I hope I have conveyed what the adhesions are, how they are made, and they're important. So if that is unclear, we should discuss, because that's the main thing. Next is you're excited about adhesions and in your project and say, okay, I'm going to study focal adhesions in this project, but what? And because I already also said there are a bunch of proteins in these focal adhesions. There is the receptor ligand bond, and then in the middle, there is maturation proteins, and they also attach to the actin. If you decide to study focal adhesions, which protein should you kind of order the antibody for, right? There's so many of them, and you start reading, and different papers will study different ones. If somebody will say, okay, I'm going to study focal adhesions by staining for vinculin. But why vinculin? Why not paxillin or zixin? And why not just do integrin labeling? Or why not talin, right? Or alfactin? I would strongly encourage you to read this paper. This is a while ago, but it's a fantastic paper sort of categorizing these proteins. And that will kind of inform your choice of which one to study. So overall, there are three classes. One is these first ECM binding proteins, then there are these core proteins, and then these actin binding proteins. So actin bind, if you're more interested in how the adhesions are related with actin dynamics, then you should be looking at alfactin and vinculin, talin. There are others as well. These are the popular ones. However, if you're really, the goal is to study focal adhesion maturation, without core proteins, there is no maturation, because that's where the stabilization of focal adhesion happens. So these are the core proteins. So paxillin, zixin, focal adhesion kinase. So earlier, when people stained for actin, vinculin, and phospho-Fac, then they were basically going for all three. So how does the actin relate to focal adhesion? Whether the adhesion is mature, they're core proteins, meaning focal adhesion kinase, and whether the adhesions are bound to actin, that's why they stain for vinculin. So this sort of should inform which proteins to kind of concentrate on. Last one is ECM binding integrins, and they are different for different alpha beta subunits for different types of ECM proteins. Collagen 1, collagen 4, fibronectin, laminin. So they all have sort of their own sort of integrin subunit that responds to them. So how that, this is a very detailed paper. I just took out these two plots, which I thought were interesting and clever. So this uses sort of speckle microscopy, where these different proteins are labeled, and these little speckle and their motions are analyzed. So this is both a biologically and both sort of physically very interesting sort of measurement. So here left average speed of these speckles corresponding to these individual proteins. So integrin, Fac, zixin, paxillin, vinculin, talin, alpha-actinin. What you would see here is that the ones that are moving the least, they are attached to ECM. So that makes sense. If the protein is attached to the ECM, ECM is the most stationary part in all of this, they will move the least. The inside the cell, the glue is moving around a lot. So there, these proteins that are connected to actin, which is continuously in motion, they're moving the most. So that's one way to categorize these proteins. The ones in the middle, core proteins, are sort of intermediate speeds. So they're both attached to the ECM and to alfactin. The average coherency here is that how strongly does the motion of this protein, the colored ones, relate to the speckles that are labeled on F-actin. So how strongly are they correlated? That is one way to categorize as well. So vinculin, talin, alpha-actin, and they are very strongly correlated. Their motion is strongly correlated to the motion of F-actin, meaning that's why they're actin-binding proteins. The farthest away integrin is least correlated to actin, so meaning that is farthest away from F-actin motion. That's how kind of one of the ways they were able to categorize these proteins. So I think it is, this one is very useful if you're interested in doing some research in focal adhesions as to which proteins to focus on. And that depends on the question that you're studying. Any questions here? You can just raise your hands or shout out, I'll stop.
I have a question.
Yeah.
So you mentioned that for catch bonds, as you pull, it'll become stronger. So a single integrin complex, that becomes more stable, right? So can I say that the number of integrins will also increase, or the focal adhesion area will increase? Or is it just the one single bond becomes stronger, so it is harder to break?
So both, I would say, is that when in catch bonds, when if there is a force applied on a single bond, actually, this is something I'm going to also talk about later. So if the focal adhesion is really small complex, then forces don't really matter that much. They have to turn into sort of adhesions which are above 1 to 2 micron square area. After that, the catch bond dynamic even is valid. Otherwise, it's not. If it's a nascent sort of focal adhesion, focal complex, then catch bonds don't do much. And that is something I'm going to talk about in about 4 slides, and we can come back to it.
Thank you.
So why do we need all these different types of bond dynamics, different types of different proteins that make up these adhesions? And I like the example of cell migration because that's what got me sort of interested in this field. Is because of this really fantastic, again, spatiotemporal diversity of different structures. So even this migrating cell at the front, these protrusions, which are driven by these actin polymerization, but there's hardly any adhesions, very tiny little nascent adhesions, because they need to be, because actin polymerization is the main thing that needs to happen. At the same time, there need to be little adhesions underneath it, otherwise they'll just be floating and go away. So here you do need very nascent small adhesions that could be turned over very quickly to enable actin polymerization. As you go back in the cell, there is a lamella here where there are adhesions need to be a little stronger so that the the front can keep exploring and keep protruding. As you go back, nucleus and go very back, these adhesions are much stronger. These are more likely to be catch bonds and strong because they will need to stabilize. Like in walking, if you're walking and sort of on a slippery surface, you better stabilize one foot, right, before you start to explore the next one. And the foot needs to be under your body. If you put it forward too much, you're more likely to slip. So the front foot is really driving the migration, but it's just not possible until unless the back foot is nicely stable. So imagine it that way. So in different types of cell migration, and again, the surface matters. Like I said, the surface is slippery versus doing walking on sand versus turf versus oil, mud. That's not attractive. But and the strategy of migration changes how much stability you need in the back foot versus the front foot. And there is a role for exploring the surface versus stabilizing the foot and the bond. So that's why you need sort of all these different types of bonds, and they all have a role to play. And if they get messed up, meaning if I stabilize the front adhesion, the cell will adapt itself and find a new front and kind of repolarize itself. If I make the rear sort of more focal complexes, then it doesn't need to stabilize, then it'll roll over like those fish keratocite examples I will talk about. So that's what gives rise to these different variations in adhesions across the cellular space, gives rise to many different modes of migration, which is again a totally different lecture. But the migration is a really fantastic example of why we need all these different types of adhesions and what roles they play. Connecting it to some more signaling pathways. So this is a slide you can look back at if you're interested in something like that. So in the front of the adhesion, there is nascent adhesions have these RAC activation, which is sort of an antagonist for Rho. Rho is generates more contractility, more forces. In the front, you don't need more actomyosin forces, you need actin polymerization. That's why RAC activation is helpful. RAC then gives rise to actin polymerization. Again, that by itself is again a different lecture, but here adhesions, nascent adhesions do this part. When there are focal complexes and focal adhesions in the back, they have a different signaling pathway because of the proteins involved in them. Nascent adhesions don't have the proteins, the ones that give rise to Rho, and that's what happens here. So like it said, FAC and Paxillin, they give rise to Rho, and Rho and RAC, like I said, they oppose each other. So here RAC goes down, so that disallows sort of actin polymerization, because now you don't need actin polymerization that much, just need enough to kind of stably make these stress fibers that give rise to forces. So it's all sort of coordinated in this adhesion continuum. In the front, there is a lot of assembly. In the intermediate phase, there's maturation and growth. In the back, there is more breakdown happening than assembly, because those are already made. For the cell to move forward, they just need to break. So that's what the catch bonds are doing. Here, I'll make a transition in terms of like what the properties of these adhesions are. So I'll provide some examples. Before I do that, let me know if you have any questions. So next, I have selected a set of experiments to kind of discuss that, illustrate some basic properties of adhesions. So let's look at this observation here. So here, there are these from this in this paper from Christian when he was a trainee, actually. There it is. There are these patterns, 10 micron patterns, 20 micron, 30 micron, 40 micron, 50 micron patterns. So patterns of fibronectin, I think. And the cells are, I think endothelial cells are placed on these micro patterns. So cell is forced to only occupy the area that is patterned. So here, if you look at it on the right-hand side, this cell is bigger, clearly, and it has more focal adhesions at the edges. The cell on the left is smaller, so less focal adhesions, more kind of diffuse focal adhesions. So the question is, does larger spreading lead to more focal adhesions, or is it just because there are more ligands, because it's a bigger pattern, more ECM? To answer that question, there's something clever that was done. Take the same amount of protein, let's say in panel A here, and distribute it over a bigger area by way of making these little tiny dots. So the bottom left column, bottom panel, has the schematic is showing the same amount of protein as the top column, but distributed over a bigger area. So this will answer the question: the cell spreading, is it making more adhesions because of more ECM protein, or is it because of spreading? So going to the right, this graph is saying is that ECM per cell is basically the same across these three examples, but the cell spreading went up because now the cell can jump over these little micro patterns and kind of spread. And focal adhesion amount really increased. So if you have this big pattern with lots of ECM, focal adhesions are smaller, lower in total, and the cell is spread more, then net focal adhesions are bigger. So what this is saying is that if the cell is bigger, there will be more stable focal adhesions. And why is that? Is something we'll kind of talk about next as well. Another sort of observation, this goes into more mechanosensing part, traditional mechanosensing of stiffness. So just observe these fibroblasts. This left is glass, 150 kPa gel, 30 kPa gel, 4 kPa gel. So right one is soft, left one is stiff, essentially. And just the cell shape, this is effactin. So on glass and stiff ECM, cell is more elongated, right? And on soft ECM, cell is more circular. So why is that? Why are stiffer ECMs leading to greater cell elongation? Like essentially, there is nothing. If I didn't show you these images, why should that happen? Why should the cells be elongated? And I have made a big deal about saying adhesions are the main sensors of mechanical cues. So the next obvious thing is to look at these adhesions. So alongside these cell shapes, let's also look at adhesions, but temporally. So left one, I should have drawn a line here, is that left of this space is 2 megapascal, 5 kilopascal. After 1 hour, both cells are kind of round. But notably, the focal adhesions are different. So on the left one, focal adhesions are bigger and sort of elongated, and the right one, focal adhesions are smaller. So even though the cell shape is round, focal adhesions have already responded. So this gives you an idea, is that focal adhesions respond to mechanical cues first, and then the cell responds. So that goes on. So this is a 6-hour video. So here cell is more elongated, and on the stiffer substrate, on the rightmost panel, 5 kPa cell is still round, and focal adhesions are smaller. Another thing to note here is that in this, from the left third panel, 2 megapascal, 6 hours, focal adhesions are pretty elongated. That kind of raises another question. Is cell elongation causing longer and bigger focal adhesions? So if the cell has already picked a direction, then it is pulling forces in that direction. And that is, of course, going to give rise to elongated focal adhesions in this direction. Or, in my mind, a more far-fetched sort of hypothesis could be is that focal adhesions decide that I'm going to be elongated in this direction, and cell sort of responds to it. So a tail wagging the dog sort of thing. And surprisingly, and I'll show you the data and then say surprisingly. So here, to answer that question, you have to again look at it temporally. So these are the videos of focal adhesions over 24 minutes, going forward to 300 minutes, to see what happens first. Do focal adhesions elongate first, or does the cell elongate first? So here, this is focal adhesion orientation, higher number, more elongation. Over time, they quickly basically are elongating. But the cell aspect ratio, which is cell elongation, higher the number, more elongation, which is aspect ratio is the ratio of major axis over minor axis if you fit the cell in an ellipsoid. So here you can see that there is a little lag. So I think the key point is, I would say 2.5 hours. At around 2.5 hours, cell is still pretty round. You can even say 2 hours, 2.5. Cell is pretty round, but the focal adhesions are quite elongated already. So this is actually showing that the focal adhesion elongation in a certain direction of cell choice, whatever the polarity is, which again is a totally different topic, that happens first, and the cell, the whole big cell, sort of responds to it, these micron level polarizations, which to me is really fantastic. And I wouldn't have predicted that if I didn't know this result, is that these tiny little adhesions are sort of dictating how the cell is going to polarize, as opposed to cell polarization dictating focal adhesion elongation. So that's why I really like this experiment. Any questions here so far? Okay, so the next example is it's also a clever experiment showing that if you now force focal adhesions to be formed elongated, which you can do by nanopatterning of these ligands, ECM proteins. So in this side, the ECM proteins are nanopatterned, meaning focal adhesion has no other way but to be elongated, because it can only form in these lines. So if you just put the cells and then allow the focal adhesions to only form in along these lines, the cell responds to it. Cell also becomes elongated. So, and this also goes to kind of the stress fibers are elongated and that are wrapping the nucleus. So nucleus also gets elongated. So these tiny nano elongations of focal adhesions leads to stress fiber alignment, nuclear elongation, nuclear squeezing. And if these experiments were to be repeated now, 10 years later, you can look at like different types of gene expressions, and even epigenetic changes in the cell may respond to these nano elongation of cells, because cell confinement leads to nucleus confinement and nucleus compression that has been shown to cause DNA damage and all sorts of different downstream responses of cells. So adhesion, physical properties of adhesions are important. Now, this experiment is sort of measurement. It's going to answer the question that was raised earlier. How does the area of the size of the adhesion relate to the force it is either generating or supporting? I should say supporting, because that's what is measured here. So on the left, it is showing that these little dots, their movement can be correlated to to forces, which is like traction microscopy. And if you simultaneously label focal adhesions, then you can also look at the area of the adhesion alongside the force that is being supported by that specific adhesion. So on the left here, in the middle, area versus force. So you can sort of appreciate the linear profile here, linear relationship. Higher the area of the adhesion, higher the force it is supporting. So that goes to something I was talking about earlier in the beginning. Meaning if the focal adhesion is really small or nascent, it won't support higher force. As it gets bigger and bigger, it will generate higher force. What does what is, again, chicken and egg problem. If you reduce the force, will the focal adhesions go smaller? That's an interesting thought. here, but this trend only holds true for focal adhesions larger than 1 micron square. That's what an important point was made in this paper, because you can see there are outliers, right, along this line. The reason is that if the area is really small, then these nascent adhesions are in this noisy sort of step of adhesion turnover. You may catch the force when it was about to break or about to form. So these nascent adhesions can show a lot of noisy behavior where force may be very high or very low, which can be the case with small bonds. So I hope that answers the question that was raised. If not, happy to discuss more. Yeah.
Can I extend the question? So the force generated here is by the myosin, like it's predominantly the active force, right? Or is it also the passive mechanical, like the springs, the stiffness of the cell and stuff? And the other question was, when you mentioned area, is it the area of the single focal adhesion or the area of the cell?
It's the area of a single focal adhesion. And the force is, generated by the cell. There is no external force.
Okay, so that means that if the cell is elongating, so the focal adhesions have been elongated in one direction, just because of the fact that the focal adhesion on the sides are weaker, that's why the cell like sort of elongates in one particular direction, right?
Yeah, that is one of the reasons. Other could be that if the focal adhesions are elongated in one direction, then the actin fibers have no choice but to elongate in that direction, and they form in that direction. And then in terms of polarization, if the cell decides it's front and back, then there are more protrusions in the front, there is more rack activation. In the back, there is more rho. So the rho rack gives this polarization and gives the cell its sort of dipole, okay, go that way. And so there is, in terms of polarization and elongation, the coupling of these chemical sort of gradients within the cell and the physical gradients of focal adhesion sizes and elongation, they both sort of couple with each other. Hope that makes sense.
Yeah, thank you.
The other is this, I also like this experiment is because this sort of is the most, I was going to say the only one, or at least the one I remember the most, answers that question, is that do forces cause higher adhesions or higher adhesions cause forces? Like I was saying, it's a hard problem to decouple. So this experiment, what it did, again, from Christian's lab, is that these are these micro posts made of PDMS. Excuse me. And in what they did is that in some of the posts, they added these magnetic nanowires. And so there'll be one post here and there that on which you can apply a magnetic force. So like a magnetic tweezer type of situation. And now, if a cell is laid out like this, is spread out, and it will autonomously form some adhesions on different. So this is one of the projects I did when I was a PhD student with Christian's lab. It was a modeling project looking at what kind of physical principles would explain this higher focal adhesions at the edges, and something I'll discuss in a second. I should have included here, but I didn't. So there we were able to figure out how these focal adhesion maturation thermodynamics couple with actomyosin contractility to give rise to these distributions of focal adhesions being higher at the edges. But the point here is that if now these focal adhesions are already made, now if you apply magnetic field, only one of these adhesions are going to move because of the nanowire underneath one of them. And if that focal adhesion increases its size or intensity higher than the other ones, meaning force is directly affecting focal adhesions. So that's what the plot in C is showing, is that if you do no actuation of the magnetic post, focal adhesion area is such. But if you do a single actuation, then focal adhesion area of that post on that post is much higher. So about 20% or so, right? So that is the very clear evidence that force applied, even if it's externally on that adhesions will mature. Okay. This is focal adhesion area per post after multiple actuation. This was single actuation, this is multiple actuation. So if you keep doing it, then there is a further added advantage. And I don't remember exactly how, is there a threshold if you do it 5 times and 50 times, is there a difference? I don't remember that part. So that is, that kind of shows the direct relationships that the focal adhesions respond to forces, not just govern them. Okay, changing gears a bit, unless there are questions. Like I said, I started off in the field of mechanobiology with modeling of these processes, contractility and adhesions back in 2005 to 8 or something like that. So long time ago. But I'll give you some kind of thoughts about thinking at that time. And honestly, a lot of it hasn't changed since then. It has been evolved for sure, but there are some basic principles that we can discuss. There's one very simple model here, which actually came much later, but I appreciated it in 2011. And I was working on these things several years before it. But this is a very, I think, good example. I generally like simpler models that explain the same process better than making it too complicated. And this is an example of a simple model that is, I think, very strong. So on the left-hand side, the basic goal of any model is no model can capture everything. And that's why all models are wrong. But what are we trying to capture, and what simplest notion can capture that observation is something to think about. So in this one, the idea is that on soft ECM, focal adhesions are smaller. On stiff ECMs, focal adhesions are bigger. What is the simplest explanation for this? And if you stain for a bunch of proteins and all that, you can come up with a complex story around it. What is the simple physical explanation for this? So here's one idea. On the left, whenever let's say the bond is formed, and you apply a force, if the ECM is soft, it's going to deform more than the stiff ECM. Because the ECM is going to deform more, this distance between the cell membrane and the soft ECM may increase. And thus, the probability of this receptor, catching the ligand will reduce because of the increased distance. So that will reduce the binding rate of focal adhesions. On the right-hand side, even if you apply the force, the ECM is not going to deform that much. So the spacing between the receptor and ligand doesn't really change that much. So binding rate is going to remain very strong. Now that you have kind of made that theoretical concept, which you can do with a pen, right? Once come up with an idea, the next thing, if you're a theoretician, you would try to put them into equations and connect them with known sort of processes. So there is this binding rate, bond dynamics process that has been shown before. So you have a binding rate and unbinding rate, and there is this K, which is the stiffness of the ECM, Z is the deformation. Here, if the deformation, because of the minus sign, if the deformation is low, then the binding rate is high. If the deformation is high, the binding rate is low. Exponential is because of this 1978 paper, Bell's law, basically, bond dynamics, and they've shown these bond ruptures happen exponentially. So that's why the exponential function. And if you then connect simply forces and the deformation with the spring constant, you can show higher forces will lead to higher bond dynamics for a given stiffness. So that is one model. Basically, there are 3 variables, 2 variables, and 2 binding and unbinding rates. So that explains why stiff ECMs will give you higher focal adhesions. Backtracking, this is something I was working on during my PhD. So there we were looking at more thermodynamic principles of bond formation. So on the left, there are these integrins, which is known. They're low affinity integrins that don't make a bond, high affinity integrins in the straight state. They do make a bond. They're alpha and beta subunits. So let's say as these bonds are being formed and unformed, they maintain thermodynamic equilibrium between the 2 states. Mu is some kind of a potential assigned for high and low affinity integrins. This psi is their concentration. There is some reference. And let me finish this here. So this is the potential energy stored in the closed bonds. So let's see on the right-hand side. So there is a thermodynamic sort of state here, where if you apply a force on this bond, Fi, then there is a delta, which is the stretch that is going to be the work done on the bond, and that is going to reduce the energy, right? And as the energy is reduced, needs to be balanced. So the only way to balance on the right side is by increasing psi h, which is the number of high affinity integrins that are bonded. So to compensate for increase in forces, they need to be higher concentration or higher, needs to be conversion from low to high affinity integrins, and that will give rise to focal adhesion formation. So this was sort of a principle of thermodynamic equilibrium giving rise to focal adhesion clustering. Whether all of these terms exactly kind of are validated, no, but it gives a sort of a physical explanation for why rise in forces, almost a counterintuitive mechanism, which is seen experimentally, why rise in forces gives rise to higher adhesion clustering. So if there are any questions about that, I'm happy to kind of discuss as well. And in another model, we also connected these three things. So there is the ones I was talking about. Much of the evidence for it wasn't there still in those early 2000s, mid-late 2000s, but we kind of, based on the information we had, we connected these three things. If there is a focal adhesion formation, that will give rise to a source of contractility. Here it is calcium ions, but also Rho GTPases that will give rise to forces and so on. So this is a feedback loop, mechanosensing feedback. I will then change gears into 3D and cell adhesions, but before that, so this was the basis of animal cell adhesion, what they're made of, how they work, where does the mechanosensing come from, what are the main proteins. And again, the adhesion literature is a lot. So before I move to that direction, I want to see how much time I have and are there any questions so far? So Annie or Becky, should I just keep going? How much time should I take?
I think you should probably wrap it up because it's been an hour.
Okay. So yeah, like 5 minutes or 2 minutes?
5 minutes is fine. Yeah, I wouldn't go
Okay.
over.
All right, sounds good. So I'll so there is a lot more to it. I'll go through it quickly. Just to kind of, if you're in, so this is all, whatever I said mainly applies to a bond is formed and then how is it stabilized, what happens within the cell, what are the sort of downstream pathways. And a lot of it seems like it is only applicable in 2D surfaces, but in 3D, of course, it's going to be complicated. These schematics are showing that, okay, if there is, imagine a rope, and if you pull on it like this way, perpendicular, then you get no resistance at all, right? And if you pull along the rope, you get a lot more resistance. The rope didn't change; its mechanical property is still the same, but the direction of force application gives you different types of resistance. And you can imagine that will happen in 3D collagen fibers. If an adhesion is made, but the force is along the fiber, the adhesion will think, oh, I'm on a very stiff surface. But if the force is perpendicular to the fiber will just buckle and the adhesion will think I'm on a very soft surface. So within a same environment, the different adhesions will think I'm in different soft and stiff surfaces. That makes for a much more complicated sort of adhesion dynamic. And so I just wanted to throw it out there. So if you see results in 3D, they may be different from 2D, likely will be, but for very different reasons, not just the confinement, not just collagen fiber thickness or concentration, but just by force vectors, right? Being perpendicular, tangential. So that is one thing to think about. In multicellular, which my lab does a lot more than kind of single cell adhesions, I would encourage you to think about this differential adhesion hypothesis, which gives rise to sorting of cells in development and diseases as well. Now, the basic idea is that different types of cells like each other differently or not so much, just like people, right? So same type of the cells that like each other, they will have stronger cell-cell adhesions and weaker cell matrix adhesions. They will coalesce around themselves. And if that happens, they will generally do that first, and that will become the core of a heterogeneous population. And the cells that are not too much into each other, they don't make strong cell-cell adhesions, they will be at the periphery, but they have stronger cell ECM adhesions, and they sense the environment more than they sense each other. So here, these bud cells and these other cells, liver cells, are kind of one example of that observation in this 1975 paper. These liver cells come in the middle, they sort themselves because they're more cohesive, and the ones that like the cell matrix adhesions more, they're at the periphery. And there is a certain set of experiments which I won't talk about. It's just 3 slides, but you can look at this paper that kind of first I made a big deal about that hypothesis, but then I say that's not it. The same thing can be said about contractility. If there are certain types of cells that have higher forces, they may come in contract in the middle of a heterogeneous population, and that generate lower forces, they may be at the periphery. Are the forces and adhesions kind of cooperative or antagonistic in this case? Not clear, but in heterogeneous populations, both things can happen. They may be differential adhesions or differential forces. Finally, this is the last slide. You can play this a little sort of game sort of thing, is that if there is cell type A, cell type B, and there is an ECM, you have these 5 combinations, permutations, I say, is how much cell type A likes to be with each other versus B, versus A and B, versus B with the surface, A with the surface. Each of these are different types of adhesions. So you have 5 types of adhesions with 2 cell types and a surface, and they will have their own rankings of affinities. So if, let's say, the cell type A likes each other the most, and cell type B likes surface the least, you can rank it. This is the predicted outcome of what the final population is going to look like. And you can keep doing this. I'll share the slides with you, and you can look at it more carefully. Of course, we can't do it right now, but just by ranking these different affinities of adhesions, you may get totally different outcomes of how sort of a micro tissue or tissue looks like, whether it is spread or it is balled up, or whether it is scattering, or whether it is coalescing onto each other. And you can imagine these different types of adhesions are happening during developmental processes, or forming organs, or forming different diseases. So that puts into context the importance of adhesions and a variety of things that we study. All right, that's it. And this is the slide I think I was asked to put earlier. These are some questions put for thought, but I think we raised a number of questions along the way as well. So I'll just leave it here and stop for questions.
Any questions?
I have a question, correct?
Okay, go ahead.
So the myosin generated force on the focal adhesions, I think you mentioned a biochemical feedback model where more focal adhesion will lead to more myosin generated force.
Yeah.
Can you name that the pathway? Like, can you give some more details about like how that happens?
Yeah, so we have kind of talked about in 2 different ways. There, one that I had done, published as a PhD student, was through the calcium ion. So there is, so with the focal adhesion signaling, there is a downstream, there's PLC gamma and IP3 molecules that attach to these the endoplasmic reticulum gates that open up, then calcium outflux happens, and because of the calcium signal upregulation of calcium ions, that gives rise to focal adhesion active myosin contractility, because now the contraction, there is a higher rate of contraction and active myosin binding. That is one pathway, and because of those forces, there is more engagement. If you can email me, I'll send you that paper. That I would say most directly addresses this question. But there are other pathways as well. I think I had a slide about Rho GTPases. So there is downstreams because of active myosin, because of adhesion signaling, there is higher Rho GTPase signaling activation, and then that gives rise to active myosin contractility. There is also some ion channel opening as well. So membrane tension, membrane stretches, like piezo is one, and then there is influx of these ions, calcium ions, and that can also be considered a mechanosensation feedback.
And can I say that like the cell-cell adhesions, like the cadherin bonds, they are like very similar to integrins, and they the kind of mechanical pathways as well chemical pathways are very similar to that?
Not really. I mean, that's a good question, but to ask. But I would say none of them are very similar to each other, and even the basic principles of bond dynamics sometimes don't apply. Meaning, are all bonds going to respond to this Bell law rupture force? Not really. So short answer is not really. The different proteins are involved, different downstream signals. In fact, there are some papers showing a crosstalk, meaning if there is higher integrin signaling, that reduces coherent bonds, weakens coherent bonds, and the other way around.
Oh, thank you.
One more question, or at least one more.
Hi, yes, thank you for the talk, loved it. My question actually goes back to whenever you were first talking about the mechanosensing of everything, and you were mentioning like the actual like focal adhesions forming. So would you say like mechanosensing happens like when the actin attaches to like the focal adhesion, or do you think it happens before that's actin attaches?
Yes, that's a good question. And I would say in two parts. One is that in the beginning, I would say in the beginning is that when the ligand sort of forms, makes a bond with the receptor, at that point also, because of soft and stiff ECMs, or if there is a viscoelastic ECM, nascent adhesions can come together just by different ECMs, and that is a small version of mechanosensing. But the big mechanosensing only happens when actin attaches to the focal adhesion bond. So the answer is mostly yes, that when actin attaches to the bond, that's where the source of mechanosensing is. But even before that, there can be ways that focal adhesions may form differently based on different surfaces.
I got it. Thank you. And then I have one more question. I was a little confused on slip versus catch bonds. So like, would you say like the biggest difference between the two is kind of just the amount of time that they have? Like the longer a slip bond is formed and it kind of becomes a catch bond, or are they two separate?
Yeah, that's a great question as well. If you're right, if the slip, so the basic property of slip bond is it shouldn't last very long. So if it forms just enough time for actin engagement and response, but then it turns over, and because of turnover, the new ones are formed. But yeah, the same slip bond, it lasts longer, then it will convert into clutch bonds. And that's what really happens in cell migration. At the front, the same part of the cell membrane makes a slip bond, but the cell is now moving forward. Those slip bonds then turn into clutch bonds, and they become the rear of the cell.
Thank you.
Yep, you're welcome.
One quick question. The question is, when cells are rolling over a surface and breaking the bonds between the integrins and the ECM, is there corresponding breakage with the actin filaments with the focal adhesion inside the cell as well?
Can you say that again, like inside the cell, meaning?
Yes, when the cells are rolling over a surface, you say that there's part of the cell membrane that is peeled off with the rolling. So the question I'm asking is there corresponding breakage between the actin filament and the focal adhesion in the cells, as there is a break between the integrins and the ECM? Or the bonds maintained?
Yeah. Yes, or?
Are the bonds maintained with between the focal adhesion and the actin filament as they break away from the ECM?
So bonds are formed near the ECM first, and inside the cell, it is just clustering of focal adhesion proteins. So that's what I would say. So it is happening on both sides. So at the bonds that are near the ECM that also come together, stabilization is sort of inside the cell, and actin bundle is also inside the cell. So I guess the bond stretches into inside the cell as well. I hope, does that answer your question, or did I not get it?
Yes, both ways.
Any other questions? Oh, one more? No, okay.